Welcome to Neuroscience Updates, presented by Naidi Bahradian. I'm an electrical engineer who has passion for neuroscience. Within this video, you are going to hear about the latest updates of neuroscience. The first news in this video, we're gonna talk about researchers who developed open source EEG visualization tool. Researchers at UT have developed a free open-source computer program that can be used to create visual and quantitative representations of brain electrical activity in laboratory animals in hopes of developing countermeasures for opioid use disorder. In the paper, the researchers described the steps they took to create a multi-tiered spectrogram for EEG analysis with an accessible and user-friendly code. They validated the program through analysis of EEG spectrograms of mice that had received different opioid treatments. Our second topic is dendrites filtering neurons' excitement. This paper found out that the Purkinje cells can modulate and filter incoming signals. The findings shed new light on learning mechanisms within the cerebellum. In mere milliseconds, trillions of chemical reactions ignite signals that travel across the billions of neurons in our brain. As we go through our daily lives and absorb new knowledge, these neurons begin to modify themselves and change their signaling properties. However, the mechanisms of how signals are integrated into the neurons to establish such flexibility, also known as plasticity, remains elusive. Journal of Neuroscience reports that Purkinje cells, the primary output neurons in the cerebellum, have the ability to modulate and filter incoming signals. The findings bring new insight into the learning mechanisms of the cerebellum and the brain. It is thought that the plasticity of these Purkinje cell dendrites is the basis for cerebellar learning. However, validation of this hypothesis was difficult due to the challenge of measuring signals within a single cell. Thankfully, in a prior study, Otsuki was successful in measuring the electrical activity on dendrites of a single Purkinje cell utilizing the patch clamp method. To measure how electrical signals travel through the Purkinje cell membrane, Otsuki measured the spontaneous synaptic activity between the dendrite and the soma, which we know is the cell body. What did Otsuki found? Otsuki found that the signals coming from dendrites far away from the soma, known as distal dendrites, were not being registered. This suggests that the dendrites have a mechanism that limits electroconduction, and that individual branches can choose whether an input passes through or not. In fact, the same signals were registered when they came from proximal dendrites, the ones closer to the soma. After further analysis, it was found that these distal dendrites modulated their incoming signals through intrinsic plasticity associated with the downregulation of an ion channel called SK channels. One of the reasons for this new finding is because similar experiments used cesium ions in the intracellular fluid, so the phenomena itself could not be observed at all. That's what Otsuki stated. Otsuki hopes to further verify this result and determine whether similar findings can be obtained with animals other than rodents such as fishes and reptiles or high mammals. This is a picture of a Purkinje cell. My last topic for this video is neuroscientists developed model to identify internal states of the brain. The machine learning model may be widely adopted to identify connections between neural activity and natural behavior. Imagine an attractive person walking toward you. Do you look up and smile, turn away, or you approach but you avoid eye contact? The setup is the same, but the outcomes depend entirely on your internal state, which includes your mood, your past experiences, and countless other variables that are invisible to someone watching the scene. So, how can you, as an observer, decode internal states by watching outward behaviors? They investigated fruit flies with fewer behaviors, and one imagines fewer internal states. They built on prior work studying the songs and movements of Amurus, Drosophila, melanogaster males. Their previous work was able to predict a portion of singing behaviors, but by estimating the fly's internal state, they can accurately predict what the male will sing over time as he courts a female. Their models use observable variables like the speed of the male or his distance to the female. The researchers identified three separate types of songs generated by wing vibration plus the choice not to sing. They then linked the song decisions to the observable variables. The key was building a machine learning model with a new expectation. Animals don't change their behavior at random, but based on a combination of feedback that they are getting from the female and the state of their own nervous system. Using their new method, they discovered that males pattern their songs in three different ways, each lasting 10 to hundreds of milliseconds. They named each of the three states as follows. The first one, close, they name close when a male is closer than average to a female and approaching to her. The second one is chasing, when he is approaching quickly. And the third one is named whatever, when he is facing away from her and moving slowly. The researchers showed that these states correspond to distinct strategies, and then they identified neurons that can control how the males switch between strategies. This is an important breakthrough. Scientists have anticipated that this modeling framework will be widely used for connecting neural activity with a neural behavior. Thank you for watching the video. If you like the video, don't forget to like and follow my page so that you can get other updates in the future. Thank you.